Welcome to ASRS's Journal of Vitreoretinal Diseases Authors Forum. I'm your host, Dr. Timothy Murray, Editor-in-Chief of JVRD. On each episode of the JVRD Authors Forum, I will interview innovative retinal researchers on their studies featured only in JVRD and how these studies will impact our patients' care in our clinics. Tune in to hear directly from investigators about the clinical implications of the newest and highest quality research in the field of retina. Welcome to JVRD's Authors Forum podcast. I'm pleased to be joined today by Dr. Marta Stevanovic, Vitreoretinal Surgical Fellow at Mass Eye and Ear in Boston, Massachusetts. Marta, you put together with your team an excellent review of intraocular inflammation, safety, events, and outcomes, and looked at a variety of anti-VEGF agents and unique agents to look at that safety profile. I'm curious, when you looked at those, how did you choose the agents to include, and how did you choose the study design?
Well, first of all, thank you so much for inviting me here. And yes, this was a great team effort study. So the way that this study came about is this is when there were being reports of Brolucizumab causing a lot of intraocular inflammatory events. And so what we wanted to look at was, how does Brolucizumab and really how do all the anti-VEGF agents compare to each other in terms of the relative risk of inducing these intraocular inflammatory events? So what we ended up doing was we did a network meta-analysis, and we looked specifically at phase 3 registry trials. So we took data from phase 3 registry trials in which anti-VEGF agents were used to treat neovascular AMD. And we wanted to answer that very question: how do these different agents compare to each other? And what we ended up finding, I'd say the most important finding of our study, is that the commonly used agents, so Aflibercept, Faricimab, and Ranibizumab, when we did our pairwise comparisons, really there were no significant differences in terms of their risk of inducing intraocular inflammation, endophthalmitis, vasculitis, choroiditis, and retinal artery occlusions.
So, Dr. Stevanovic, when you guys looked at this, one of the things that I found interesting is you used the published clinical trial data, which is the gold standard, of course. But as we know for Brolucizumab, that drug went through phase 3 clinical trials and was approved by the FDA. And it really wasn't till that drug moved into real-world usage that we identified the major complication. So, do you think there's a difference between clinical trial data analysis and real-world usage? And how does that impact a study like yours?
Yeah, I mean, I think you bring up an excellent point. So certainly clinical trials are the gold standard in terms of the data. Specifically for Brolucizumab, when the data was looked at again, it was found to have higher rates of intraocular inflammation. So that's one thing to keep in mind specifically from the clinical trial. But certainly in the real world, you have patients who are maybe receiving one medication and then that doesn't work, then they go to another one and then they're transitioned to yet another one. And those patients are, perhaps different than the patients from the clinical trials who are treatment naive. So yes, there certainly is a difference. But specifically for our study, we wanted to have the highest quality data possible going into this meta-analysis. And so that's why we chose to focus on the phase 3 registry trials as opposed to, expanding the data pool further.
And I think that was a very reasonable scientific choice on your part in design. I did find it so interesting, though, that it really took real-world experience to have us go back and re-look at phase 3 clinical trial data to see the issues that were there. So one of the things is that, we have small numbers of patients that are treated, and we look at them in a very specific way, and we're very defined about what our inclusion and exclusion criteria are. So that's the gold standard. I agree. And I think the take home that you alluded to, which was of the major agents that are in current usage, there is no real significance in clinical differences in terms of safety profile for these medications. I love that, because as a busy clinician, that's really what I want to know before I make a choice.
Right, absolutely. I agree. And kind of, maybe subjectively you kind of sense that, but it's good to have the data to back that up and to say, hey, these agents, they are similar in terms of their risk profile when you're treating patients with neovascular AMD.
And I do think that because of our experience with Brolucizumab, we look at all of our patients that are getting anti-VEGF treatment in a much more focused way, potentially differently than we did before. I think many people were unaware of the fact that if there's pre-existing inflammation in an eye, that was a contraindication to re-inject. And that inflammation in a lot of cases was anterior. So for many offices, the lack of a slit lamp delivery system or anterior segment photography may put you at a slight disadvantage to re-injecting an eye with pre-existing inflammation. What would you tell your clinician that's using these drugs in the real world? Any pearls for us based on what the study has shown us?
Yeah, I mean, I think, again, these three kind of the ones that are used commonly have similar risk profiles. And I think, uh, they're all, they're not exactly comparable, but I think they're all reasonable choices. And so that's what I would say to the clinicians who are using it.
And do you think when you see a trial like yours where you've done meta-analysis, do you think that should, is there a reason after that to go and do something that's a more real world analysis and maybe in a broader population or a different population?
Yeah, absolutely. I think, like I alluded to previously, the clinical trials have pretty stringent exclusion and inclusion criteria that don't necessarily mirror what we're experiencing in the real world. So I think absolutely applying this analysis to different patient populations is important. And not just different patient populations, but different diseases. So specifically looking at diabetic retinopathy and other anti-VEGF responsive conditions. And then also, how do biosimilars fit in with all of this? So yes, there's definitely room to expand this type of analysis and look at different patient populations and diseases.
And I think the comment on biosimilars is really fascinating. I know that's been a concern of so many of our clinicians. And we know that the FDA looks at biosimilars very differently than it looks at a novel agent. And the criteria that they use are very different. Some of us have felt those criteria are much less stringent. Do you have any commentary about that, or do you have concerns about the use of biosimilars?
Yeah, I mean, I think, you bring up a good point that they're biosimilar, but they're not exactly the same. So I definitely think it would be important to apply this type of analysis to the biosimilars, because at the end of the day, we don't really know until we actually look at the data and do the studies. So I absolutely think it'd be very important.
Right. And, it goes back to when we started using anti-VEGFs and we were concerned about, arteriovascular or cardiovascular events with these medications. And it was like, this one's better or this one's worse. And I think as time has gone on, most of us feel that kind of has washed, much like the concern for inflammation.
Yeah, absolutely. I agree. I mean, fortunately, I would say these events, they are very rare, but they unfortunately do occur. So it's important to understand, what the risks are of all the medications that we're injecting. But yes, as you said, with some of the systemic adverse events, they're not, maybe not quite as common. But I do think we do see these intraocular inflammatory events. And
So the meta-analysis that you guys did was so well structured. I think that's one of the keys to this. So we've chatted a little bit about, top end commentary and results, but there's a lot of detail in the manuscript. So I would really recommend that for someone to understand better how this was done, that we direct them to the paper.
Yes, absolutely. Thank you. It was a lot of hard work and a lot of detail, and the analysis was, very, it was a team effort. So I definitely agree with Dr. Murray. Please read our manuscript for more details and more information.
Well, Dr. Stevanovic, thanks for joining us on our Authors Forum podcast. I really appreciate that. You've given us some significant insight into what led to the paper and how it was interpreted, and really some good clinical pearls for our audience about how that data can be applied in a daily basis in our clinics. So thank you very much for joining us.
Thank you.
Thanks for tuning in to the JVRD Authors Forum. You can watch and listen to more episodes on the ASRS YouTube channel and on popular podcast directories, including Apple Podcasts and Spotify. Visit www.asrs.org/JVRD forum on the ASRS website to learn more. See you soon.